Hi, I'm Jay Rangan, and this is my presentation, TGF-Beta, SMAD2, SMAD3, SMAD, and SMAD4 Pathway by Jay Rangan. And in this TGF signal pathway, it describes all the type 1 receptors and type 2 receptors and how the TGF-beta superfamily ligands bind to a type 2 receptor, which recruits and phosphorylates a type 1 receptor. And this results in how the type 1 receptor then phosphorylates receptor-regulated SMADs, which can now bind the COSMAD SMAD4. And then the R-SMAD and COSMAD complexes accumulate in the nucleus, where they act as transcription factors and participate in the regulation of target gene expression. And then the TGF-beta signaling is present in the control of cell proliferation and survival. It acts as a tumor suppressor, and TGF-beta family contributes to resisting apoptosis, evading growth suppressors, and enabling replicative immortality, hallmarks of cancer. And TGF-beta is a transforming growth factor signaling pathway which regulates embryogenesis, cell homeostasis, proliferation, differentiation, and death. The transforming growth factor beta proteins are cytokines that are critical during the development and homeostasis of somatic tissue and whose downstream signaling regulates tumor progression. The pathway and its components are deregulated in many cancers. In the normal cells and early stages cancers, TGF-beta signaling suppresses tumor formation by inhibiting cell growth and apoptosis. But as cells become resistant to these anti-growth effects through acquiring mutations in the TGF-beta and its downstream effects, the TGF-beta signaling can act as a tumor promoter. And the TGF-beta family is broadly divided into two subfamilies, TGF-beta ligands and bone morphogenic protein ligands, based on their homology, receptor binding, and the activation of the SMAD family of transcription factors. The TGF-beta signaling is induced by many ligands, serine, threonine, protein kinases, binding to its cognate cell membrane receptors. The cell membrane receptors are classified as type 1 or type 2 receptors. The type 2 receptors are constitutively active. Upon ligand binding, they are brought into proximity to type 1 receptors to phosphorylate and activate them. And the R-SMADs consist of SMAD1, SMAD2, SMAD3, SMAD5, and SMAD8-9, and are involved in the direct signaling from the TGF-beta receptor. The SMAD4 is only known human co-SMAD and has the role of partnering with R-SMAD to recruit co-regulators to the complex. Finally, the SMAD6 and SMAD7 and we have the TGF-beta pathway signaling through the TGF, and then now we have the TGF signaling pathway drug targets, or the drug target ligand activation blockade, and alpha-GARP and alpha-integrin. And we have the drug target small molecule kinases, which acts like an inhibitor. An example of this is galunasirtib. It helps to inhibit this site. And some questions I have for this presentation is if SMAD signaling is defective, does it affect TGF-beta signaling pathway? 2, which SMAD is the only known human co-SMAD? And 3, what is the TGF-beta type 2 receptor? And 4, how many TGF-beta type 1 and type 2 receptors are known to exist in mammals? And 5, when the receptor ligand bind occurs via local action, what is the signaling called? And here's my works cited resources. And thank you.